Okay, good evening everybody. It's a great pleasure to actually welcome so many people to such a well turned out lecture this evening. Particularly like to welcome Nick's family and friends, some of whom have travelled quite a long distance to be with us tonight. It's a great pleasure to welcome you, as well as external academic visitors academic colleagues who come from, again, quite a long distance. Just a little bit briefly around the role of inaugural lectures. They partly give the chance for a newly appointed professor, whether that's someone like Nick, who's been appointed through our internal promotions process, or whether someone who's been appointed externally as a chair, to give a talk around their area of research expertise. And it gives an opportunity to raise the profile of research at Keele in whatever discipline, in this case in science and technology and medicine. So I'm just going to give a brief introduction to Nick, a biography of Nick. So he did his undergraduate and PhD in Glasgow. His PhD was at the Beatson Institute of Cancer Research in Glasgow. His supervisor is here this evening, so a great pleasure to welcome him. He then went on to carry out two postdoc periods of research. Firstly, he spent 4 years at University of Texas, Southwestern Medical Center, and then he returned back to the UK and for a further 2 years was at the University of Edinburgh's Roslin Institute, where he studied human embryonic stem cells and their potential application in orthopedic tissue engineering. And following that, Nick was appointed to a lectureship in stem cell biology at Keele in 2006 in the Institute of Science, Technology and Medicine. He was promoted to a senior lecturer in 2009, to a leadership in 2013, and then last year to professorship. At the same time, Nick also became director of Keele's Institute for Science, Technology and Medicine last year, having previously been the research lead for regenerative medicine in the institute. As you'll have gathered from Nick's title, his research is in the area of stem cell biology, which is a field which has seen enormous developments over the last decade or so. And Nick's made a significant contribution to that. It's an international research profile, over 80 highly cited papers. He's averaged over £250,000 a year in research income since coming to Keele, and he's recently led a successful bid for just under £500,000 for a Confidence in Concept award from the Medical Research Council. He's also successfully engaged with industry, including 3 patents, 2 of which have been licensed, and holds a number of consultancy roles. And he played a pivotal role in the first in-man cell therapy trials at the University Hospital of North Midlands, targeting ischemic stroke. I think something that I'd like to particularly mention around Nick is his commitment to postgraduate research supervision. He currently supervises 6 PhD students and has supervised a lot of PhD students in the past. And again, in terms of growing Keele's research activity, having that number of PhD students is a real credit to Nick. He hasn't exclusively done research. He has also, for 5 years, been the course director of the MSc in Cell and Tissue Engineering, leading modules in stem cells, in cell and tissue engineering, and developmental biology. And I think probably the final thing I'd say about Nick is his significant international dimension to his work and his activities. So he has multiple overseas research collaborations. He's acted as a transnational ambassador across several countries and held visiting professorial positions at Sichuan and Guangzhou universities, 2 of the leading Chinese universities, and also at Boku in Vienna. And probably the final thing is to say that Nick led the very successful general engineering submission to the last 2014 Research Excellence Framework, in addition to, as I mentioned, taking on the wider research leadership role. So increasingly moving into research leadership, as well as being a very successful researcher in his own right. So can I therefore formally welcome Nick Forsyth, Professor of Stem Cell Biology, and invite him to present his inaugural lecture, Stem Cell Biology, Going Back to the Future to Improve Tomorrow's Medicines.
Thank you, and thank you, Mark, for the kind words. And also thank you to the audience. A lot of you have travelled a long way, so I'm very grateful. And it's a challenging thing to present an inaugural lecture, because essentially what we're trying to do is to weave two stories. We're trying to weave a story of me and my research career, but also trying to weave in something about the research that I do, and hopefully come out with something coherent at the end of that, which allows everybody to take home some clear messages. I would also just like to say thank you for all the nice comments in relation to the suit. It's not often I wear a suit, and I do appreciate it. Okay. So the title of the talk that I'm going to present is Stem Cell Biology, Going Back to the Future to Improve Tomorrow's Medicines. Now, I didn't want to leave anyone shortchanged, so this is the Back to the Future bit, okay? And we're not going to reference Back to the Future, really, although we are, okay? But we'll understand that as we move through. We're interested in cells. I'm interested in cells. I study cells. I've studied cells over my academic career. And this is a little bit of Back to the Future, I guess. If we look over onto your left-hand side, these are the original sketches from Alexis Carroll's Journal of Experimental Medicine paper published in 1912. Now, back in those days, it wasn't so easy to take pictures of cells. So essentially, you had an illustrator, and this guy was Willy Schmidt, and Willy Schmidt would look down the microscope and he would record what he saw, and he would record what he saw in beautiful detail. And there's a real elegance to this approach, and he had to do it quickly. They'd be looking at moving cells going through solutions, and as quick as he can, he'd try to take a photographic plate in his mind, and he would draw this down. Now, what we do these days is we take pictures using microscopes and cameras, okay? And we can zoom in and improve the resolution as we go. Now, what you're looking at here is a picture of mesenchymal stem cells. They're a cell that's isolated from bone marrow, and we're going to talk about them in more detail as we move forward. But I think you would agree with me that there's a real elegance to this approach, and we mustn't lose track of the skills and the abilities that enabled this first report of mammalian cell culture, because this was the very first report, 1912, to 2017, so we've got 105 years from that first report to where we are today. Those were mesenchymal stem cells, and we're going to have a couple of number slides just to help frame the subject matter. Mesenchymal stem cells form the basis of the cell therapy industry, okay, and it is an industry. This is a $30 billion market. Here we have our global tissue engineering and all of its lovely numbers, and you can read these yourself. But the key thing is this one here, organ replacement transplant, is this little orange bar here that hasn't really grown and won't change. And the reason it won't change is that we have waiting lists for transplantation because there aren't enough organs. So we have to find solutions. And the solutions lie in this cell therapy arena, where we can take cells out of the body, we can grow them up, much the same way as Carroll described in 1912, with some extra sophistication perhaps, and we can apply these cells into orphan diseases, that is, things that have no cure. And there are many, many diseases that have no cure or limited treatment options. Cancer, diabetes, lung disease, etc., etc. Predominantly, however, as you'll see from this light blue bar, we have an application in orthopedics. Okay? So we have predominant application in orthopedics, but a massive industry. Now, this industry itself is underpinned by an approximately $10 billion cell culture market, okay? These are the things and the ways that we grow cells. The cell culture medias, the plasticware, the engineered equipment. So putting all of those two things together, we've got a global industry of quite significant depth and weight, okay? That is growing in all arenas that we look at. Okay, so that's the sort of frame. That's why we're looking at those cells. That's why we look at cell biology. But let's flick back. Mark gave a very kind introduction, but what I'd like to do is to try to weave that first story. So starting off with a bit more about myself. Glasgow University, where I did my honours degree in molecular biology, achieving an upper second class. Staying in Glasgow to go to the Beatson Institute, supervised by Ken Parkinson, who's in the audience, to do a PhD in molecular genetics. Now, the title of that PhD was functional demonstration of a mortality phenotype associated with 4-sen-Q23. Now, one of the things that you'll understand about science is that we talk in an absolutely impenetrable language, that if you're not a scientist is bewildering. My dear wife commented on my brief for this inaugural lecture and congratulated me. She said, I understood every single word until you got to the second sentence, and then it all fell apart for me. And that's a criticism that we wear, but we spend our whole careers learning to speak this language. So it's difficult decoding, but I've attempted to do that here. So we've decoded this thesis title into a bit of chromosome 4 that stops cancer cells dividing. Okay, so we've tried to make that very, very lay in language. So a bit of chromosome 4 that stops cancer cells dividing. So what does that mean, and what thought processes does that kick off for me? Well, cancer cells have an unrestricted growth because they lose control of mechanisms designed to stop proliferation. They grow indefinitely. Okay? Those mechanisms which stop proliferation have a key role in aging. Now, what we did during my PhD was we identified a small region on chromosome 4 that caused cancer cells to age and stop proliferation. Now, clearly, I'm not expecting you to take all of this in here. What we're looking at, this is chromosome 4. Our DNA is packaged into chromosomes, and each one of those chromosomes has different bands, and different genes are located in different areas. Well, we can use techniques to analyze which areas of these chromosomes we have and which we don't have in cancer cells. The key message I want you to take here is that each one of these little dots represents around 5 hours hard labor. Okay? Each one of these little dots is about 5 hours hard labor. Now, what we were able to do sequentially, by lots of exercises and hard labor, was define areas which we thought might matter. Define areas where we were able to demonstrate loss of areas that associated with this loss of control of mechanisms to stop proliferation. And we were able to zoom in on those areas. We were able to abandon areas because they didn't do the right thing. We were able to focus further on different areas and then zoom in further and then zoom in even further again, ending up with a little place on chromosome 4 that, when reintroduced into a cancer cell, would stop that cell growing. Now, at this point, an important lesson was introduced to me, and that important lesson reflects the vagaries of science. And the vagaries of science are that even the best idea in the world, unless you have money behind you, it gets put on the shelf, and it stays there. And this little region, this area responsible, the gene that lies in this area, remains to this day undiscovered. Maybe someone will circle back and find it, but until that point comes, this is just an example of many things which lie. Many of the best discoveries are yet to be made, but they're sitting on a shelf half done. That might be a theme. Okay. Now, the other thing with these are the 5 hours hard labor, is that nobody told me that PhDs were risky businesses and carried a grave risk of injury. So all of those 5 hours of hard labor, of course, resulted in bursitis or tennis elbow. And I had to sit with my hand elevated, holding onto a broomstick, not allowed to go into work, not allowed to run any more of those gels. So a risky business indeed. Okay. So we mentioned aging. So what is cellular aging? Well, it's complex. It's driven by multiple factors. These include stress, and ultimately present themselves as a cessation of proliferation that we call senescence. Okay? These stresses can include oxidative damage, that is too much oxygen, and either restricted or excess nutritional supply. That might be in the form of sugar, that might be in the form of serum. Now we're going to go back to the future again, okay? So back in 1987, these clever people, Derek Liu and a bunch of colleagues, demonstrated that if you took cells from a mouse and cultured them routinely, as we understand and we know, they will stop dividing after a period of around 35 days, okay? If, however, we took these mouse cells and removed one of their nutrient supplies, in this case serum, those cells would keep on dividing essentially indefinitely and wouldn't stop. Okay? And these are the cells here. This is them here in the absence of serum, and this is them here in the absence of serum. These are the guys that were cultured with serum. So what this tells us, going back to the future, is that back in 1987, there was a recognition that the way that we cultured cells might not be right, or we might be doing something that wasn't particularly accurate, that we weren't reflecting all the conditions that we needed to. Okay, so cellular aging. So now at that point, following on from my PhD, I developed a thirst in aging. So how was I going to explore this further? Well, I moved to Texas, to UTSW Southwestern Medical Center, and I studied under these two guys as a postdoctoral fellow with US Department of Defense Breast Cancer Research Fund support, Jerry Shea and Woody Wright. That was from 2000 to 2004. In 2004, 2006, I moved back to Edinburgh to study under this guy, Dr. Jim McWhirr, who we'll hear more about later. And that was at the Roslin Institute, as we heard. So we took a trip to Texas to study telomeres. So I showed you a little cartoon of a chromosome earlier, chromosome 4. Well, all of our chromosomes, all of our DNA is packaged into chromosomes. And at the end of each chromosome, we have these little structures that are called telomeres. Okay? Every chromosome has a telomere on the end of it, or rather pairs of telomeres. Every time a cell divides, it loses a little bit of that telomere until you end up having really short telomeres, and at that point you're old, and at that point you lose the ability to divide. Okay? So tracking down our little diagram here, here's telomere length, here's cell divisions. Routinely, as the cell divides, we lose telomere length until ultimately a little signal goes out that induces the cell to stop dividing. Stem cells, embryonic stem cells, sperm, eggs, intestinal stem cells, express telomerase continuously through life. That's why your skin keeps dividing. Our skin stem cells also do that. It's why your stomach keeps renewing, because your intestinal stem cells also do that. It's why your brain cells keep regenerating, because your neural stem cells do that. But most body cells are telomerase silent. So our telomeres shorten indefinitely until we hit senescence, a term we brought in earlier. Apart from cancer cells can switch back on telomerase, and then they maintain their telomere length and are then able to divide continuously. So telomerase is the enzyme that repairs telomere ends and can be used to extend cellular lifespan, indeed make cells immortal. Telomerase was first described in 1984 by Carol Greider and Liz Blackburn, looking at a freshwater microbe. And actually, this discovery has such a fundamental bearing on the research community that they were actually awarded the Nobel Prize in 2009 for this discovery, along with Jack Szostak. What Jerry and Woody did was showed that if you took telomerase and put it back into normal cells, those normal cells would essentially now start to divide forever. So we're reintroducing telomerase, fixing the telomere problem, and off they go. Now the key points here are A, and A is essentially the average time that the average point that these normal cells stopped dividing. B is where they got up to with the average lifespan of cells that had telomerase, those open triangles, at the time of publication in 1997. Now, the reason they published at this point in 1997 was because of that other wonderful vagary of scientific research, the fear of being scooped. Okay? Now, there were a number of other labs working on exactly the same topic at exactly the same time, and the first to press gets the glory. The first to press publicizes, popularizes, and gets invited to go and give the fancy talks and the nice dinners and the expensive wine, etc., etc. So that was why this was published here. But these same cells were cultured up to their approximately 350 population doubling, so all the way out, all the way out, all the way out, at which point presumably someone got bored and froze them back and they haven't been looked at since. But the point being is that these cells and the re-expression of telomerase was sufficient to stop aging and allow the cells to start dividing indefinitely. Okay, now we're going to step back to the future again, and we're going to talk about this guy. This guy's Leonard Hayflick. Now, in 1962, Leonard Hayflick was busy at the Y-Star Institute in Philadelphia, and he was isolating cells from fetal human lung alongside many other tissues. Okay? In 1965, Hayflick published a canonical body of work. And in that body of work, he described the in vitro mortality, that is the aging process that underpins normal somatic human cells. One of the cell lines they isolated at that time was called WI38. Now that aging, that mortality point, was termed the Hayflick limit. Now why this matters is that up until he described this, canon held that all human cells were immortal in culture. Okay? And there were good reasons why that was thought, but it was wrong. Okay? Hayflick demonstrated that this was wrong. Okay? Now, these were from fetal human lung, these cells that were being isolated. Now, what I'd like you to conceptualize here is that the fetus is never exposed to atmospheric oxygen, and instead is exposed to something in the order of 2%, essentially getting its oxygen from cord blood after it's passed through the placenta. Now, so we need to bear that in mind. Now, there's a terrific backstory to this, which feel free to ask me about afterwards. It involves cars, it involves theft, it involves prosecution. There's a Hollywood film in the making that hasn't been made yet. So given that these cells were being cultured, this is a flask of cells, possibly WI38. Just behind them here is a cell culture incubator. Given that these cells were being isolated at 20% oxygen, and the normal physiological oxygen for those cells would be 2%, we have to ask ourselves at this point, so is there a case for oxygen stress and cell aging? And if we kind of stop and think about that for a minute, 20% is around the strength of fortified wine, okay? For instance, Buckfast. Some of you might be familiar with Buckfast. And 2% would be a low alcohol lager, okay? Those two things, volume to volume, will have very, very, very different effects. And yet, that is essentially what we're doing to our cells in culture. So what we did when we were out in Dallas was to actually look at these WI38 cells. And we did a very simple experiment. We cultured them at room oxygen, and we cultured them at physiological oxygen, and we cultured them in the presence of an antioxidant. And what we showed was that this original Hayflick limit reflected accelerated aging driven by oxidative damage. If we cultured these cells in a reduced oxygen setting, we increased their lifespan by 50%. It was a fairly dramatic increase, and our antioxidant increased things a little bit further. Now, this was reflected, if you remember we discussed telomeres, this extended cell division capacity was reflected in a reduced telomere shortening rate. So essentially, we halved the rate of telomere shortening. So we were losing half the amount of telomeric DNA each time the cell divided, which meant that the cells were able to continue dividing for longer. So Hayflick was right. There was a limit, but he was wrong with the point of his limit, the positioning. And what this allowed to do, now that we start using our physiological oxygen condition, is it now allowed us to immortalize these fetal cells, because we've got what a component of the underlying cell culture milieu correct. If we tried to do this in 21%, the cells would not immortalize. Okay. So, meanwhile, 3 decades earlier, this very same thing, this is WI38 cells, and this is in 1% oxygen, and this is in 20% oxygen. So 30 years earlier, the same thing that I've just shown you, minus the telomere shortening, had already been reported. How can this be, you ask? You must have been able to find this out readily. Well, though this might sound strange, back in 2000 to 2004, or even late 1990s, the internet was only just getting up and running. And if you wanted to find a body of work that somebody had done previously, they had to have used the correct keywords that you were going to look for, or they had to have been popular and cited by the papers that you were looking at. Otherwise, it was essentially, you couldn't see it. It wasn't there to be seen. It was invisible. So 3 decades earlier, this same body of work, broadly speaking, had been done, but it laid out of sight, and so it was out of mind. And similarly, and this was colonies, this is isolating individual cells, and this was proliferation. So 3 decades earlier, there had been a knowledge of the importance of physiological oxygen that was subsequently forgotten about. that nobody looked at for another 3 decades until we circled back. So now hopefully we're looking at the back to the future theme and starting to get a concept of why it's important to look to the past, because things may have happened in the past that we just aren't aware of, where the right answers may lie, or at least elegant insights. So this word physiological normoxia, which I've mentioned a couple of times, what does that mean? Well, mammalian cell culture, first of all, is generally and routinely performed in air oxygen. Mammalian cells in vivo, however, are rarely, if ever, exposed to air. So we culture our cells in air, which is quite patently not the condition that they would ever see. There's a kind of crude little schematic here showing oxygen and showing different tissues and arranged. Here we have our bone marrow cavity, the eye, lung, parenchyma, liver, kidney, heart, massive range, as I'm sure you'll note. And on our, here we have blood. Paradoxically, blood has a very low physiological normoxia. And why is that? Because oxygen is complex with hemoglobin, which is carried by erythrocytes. We have mechanisms to keep oxygen out of solution to deliver it to the tissues when it's most needed. This little line down here at the bottom that you might be able to notice, this is how much free dissolved oxygen is present in blood. Okay? Very, very little. It's all complexed and carried to the tissues where it's needed. And I don't expect you to take any of this home with you on this slide, but what we'd just like you to note is that we have evolved substantial, complex measures that sense and respond to changes in oxygen concentration. And those oxygen concentrations are those that are in almost the sub 5% bracket. Okay? So we have evolved systems, and those systems are conserved from yeast all the way up to humans. Those same systems do the same things in yeast all the way through to humans, which should tell us that they matter. We don't conserve things evolutionary for the hell of it. They have to play a role, otherwise they're discarded. And I came across this phrase in a legal symposium around about a year ago, recipsa loquitur. And what that means is the thing speaks for itself. So given what we've shown you there about physiological normoxia, given what we've explained to you about the way we culture cells, at this point, the thing should speak for itself. Now, let's flick back and start talking about Leonard again. So Leonard Hayflick, here he is with his flask of cells. Okay, the cells would be attached to the base. That liquid there that you see is culture media. This is a cell culture incubator. These cell culture incubators, this actually dates from 1962, cell culture incubators are essentially the same technology as was first described in 1962. Things have become miniaturized, things have become faster, sensors have become better at sensing, but essentially they're the same. Which other technology can we stop and think of, apart from the wheel, that just hasn't evolved really since 1962? And all of these incubators provide an air atmosphere that's supplemented with biologically inert carbon dioxide, and that carbon dioxide stops the growth media that the cells are in from becoming acidic or alkali. But it's biologically inert. It doesn't do anything to the cells, biologically inert. So these incubators are not designed for physiological normoxic provision, clearly. So how do we provide physiological normoxia to cells? So here we've got an example of something that we jerry-rigged when I was out in Glasgow, out in Texas, sorry. And Anne will recall me coming back from the container store with various containers that we would drill holes in and pipe gas into and seal and all sorts of elaborate schemes. And that's here we've got a pre-mixed gas that would have 2% oxygen, and we would purge this into these little tubs and seal them with plastic bungs and then put them into the incubator we just saw. You can also get these. This is an incubator. This is a modern incubator, which you'll note looks exactly the same as the one from 1962. This can be filled up with a low oxygen gas, so with a physiologically normoxic gas. But as soon as we open the door, obviously all that gas escapes. We're back up at air, and then we close the door, and then it slowly purges back down again. Okay. Or we've got these little flying saucer type things which we put our cells and plates into, pop the lid on, and then pop this into the incubator as well. Now all of these lack control and are ill-suited for function. okay, is the harsh reality. We still use all these things, and we still use all these things with the knowledge that they don't provide us with the control that we would wish them to provide. Okay, so at this point, the Texas adventure comes to an end, and I return to Roslin. Now, the reason it says return to Roslin is that the Roslin Institute is globally famous. It's also about 5 miles south of where I grew up, okay? Hence the return. Now, we can't mention the Roslin Institute without showing a picture of Dolly. So there's the picture of Dolly the sheep, and there's Professor Sir Ian Wilmot. And in 1996, a sheep was cloned from a DNA nucleus taken from an embryonic cell. It was an embryonic cell, so it was less specialised, so it was easier to reprogram. In 1997, a sheep was cloned from the DNA nucleus of an adult cell, more specialised, so harder to reprogram. Now, the guy who injected the DNA nucleus of these cells into the egg for the animal to be cloned was Jim McWhirr, okay? So this was the guy who did the technical, the non-veterinary bit, and then he would go off to and then the vets would do the implantation, et cetera, et cetera. There was a talent to this. There was something like 350 oocytes had to be injected for this to happen, okay, to make Dolly. So this was a very laborious endeavour. Now, Jim McWhirr was also one of the first labs in the UK to gain access to human embryonic stem cells. Human embryonic stem cells are a very specialised cell. They're pluripotent, and that means that they can give rise to all the other cell types found in the body. Okay? So here we have what we can call an inner cell mass. This is adapted from Thompson et al. 1998, the first description. An inner cell mass of cells from a pre-implantation blastocyst. This is a little outgrowth of embryonic stem cells, and here we've got roughly 150 cells at this point. Now, these cells have a little magical power that normal cells do not have. You can take these cells, and in the lab, you can induce them to form tissues, to form cell types or tissues such as gut, brain, bone, cartilage, muscle, and kidney. Normal cells will not do that, okay? And this ability gives a real initiative, a real drive to the emergence of our global stem cell therapy arena. That 30 billion industry that we saw in one of the very first slides gets the kiss of life at this point and picks up, because now we have something that's highly relevant, malleable, scalable, and an industry understands those things. So this is where tomorrow's medicines now start to take shape. Now, what I did at the Roslin Institute was a few things. But the key thing I did at this point was take this physiological normoxic thing, physiological oxygen, and apply it to human embryonic stem cell culture. And what we showed in this simple and elegant paper was that by applying physiological oxygen to embryonic stem cell lines, we increased their growth by an order of magnitude. Okay? So we got 10 times more than we would ordinarily. We also showed that actually by culturing these cells in our reduced oxygen, our physiologically normoxic condition, we now saw reductions in chromosomal damage, things that caused cells to go wrong. These chromosomal aberrations, we've got highlighted one here, this little join between these two chromosomes, that shouldn't be there. That's an aberration. Those aberrations are how, for instance, cancer cells are able to mutate and evolve and lead to more malignant types of cancer. The important number here, I don't want you to focus on this too much. I just want you to note that this number here is halved, 0.32 to 0.13, and this number here is half, 0.44 to 0.16. And that means that when we're culturing our embryonic stem cells in physiological oxygen, we see half the amount of DNA damage that we would see ordinarily. Okay? So that physiological oxygen is giving us more cells, and the cells that we're getting are healthier. Now, a substantial legacy has emerged from this pivotal manuscript. It's become highly cited, and it now forms the basis of a component of state-of-the-art embryonic stem cell culture systems globally. Roslin was a productive spell for me. Okay? We achieved a lot in Roslin. We were there for 2 years. We had 2 sons, both sitting in the front row there, and produced 8 papers. So it was very short, high-impact stuff. Okay? That then took me here. I saw a post advertised for a lecture in stem cell biology. That was in spring 2006. In late April 2006, I accepted the position. In August 2006, I moved and I took up the position. And in January 2007, I finally moved to the position under the directorship of Alicia El-Haj, who with hindsight was incredibly patient with me and waited for me eventually to get myself into gear and to make the move down, which a move I'm eternally grateful that I made. This is a picture from 2010. So now we're at Kiel, and the cogs are still turning. We're still thinking back to the future. We're still thinking what lessons can we learn from the past. So we're starting to think about cell culture media. Okay, this is the stuff that we grow the cells in. Cells are grown in flasks, and here's Leonard holding a flask, or plates, and the cells are submerged in a growth medium. Now these growth mediums that we grow cells in were formulated essentially by this guy, Harry Eagle, in 1954. Okay, so in 1954, Harry Eagle formulated cell culture media, and that cell culture media remains essentially unchanged to this day. This forms the basis of many or most culture medias in common use today. And here's the table from his manuscript describing all the stuff he puts into media. And that is still all the stuff we put into media, plus some specific bespoke changes. But dissolved oxygen was not a consideration then, and it's not a consideration today, okay? So with that in mind, we then started to explore this a little bit further. Okay, does dissolved oxygen matter? Surely that media that we grow our cells in just has the oxygen that is surrounded by somehow. It just magics in there. Well, we started measuring different types of media. These two graphs show essentially the same thing. And what they show is that across multiple different types of media, we see large variations in our dissolved oxygen content. Sometimes we would have 20% in there, sometimes we'd have 10% in there, sometimes we'd have 16% in there, 12% in there. It varied from time to time, batch to batch, lot to lot. Okay? So that's point number one, is that we are in no way controlling the oxygen within our culture media. We can control the environment to an extent, but our culture media is outside our control. That's point number one. Point number two is these are our medias in common use today. Now these are acronyms, okay? DMEM stands for Delbecque's modification of Eagle's media, Harry Eagle, 1954. MEM, modified Eagle's media. Iscove's modified Delbecque's media. Delbecque's media is, of course, DMEM. So everything's hooking back to these early experiments that were performed painstakingly way back in the 1950s. Okay. So this then set grey matter in motion. We started thinking, okay, can we do anything about this? At this point, my claim to fame is that to this day, I think I'm the only person who's persuaded purchasing at Kiel to let them buy domestic household fridges. And those domestic household fridges, I set to drilling holes in the back, plumbing in gases, sealing things up, essentially creating pressured vessels that would just be in no way safe. And eventually there came a point where I thought, I'm really out of my depth here. I need to stop what I'm doing or I'm going to blow myself up. And contacted a company of engineers. And that company of engineers, we sat down, we spoke. We'd patented this already. And we sat down with this company of engineers, and essentially they built this fridge to all extents and purposes. And this fridge has controlled cooling, has controlled agitation, and a controlled headspace gas. What that means is it has the right oxygen, the right carbon dioxide, and the right air in there. And what we're now able to do is to dial in precisely the oxygen content that we want in a rapid manner. So now we can create media that's got the right oxygen on it and pop it on our cells, and then pop our cells into a condition that has the right headspace oxygen. Okay? So we had controlled cooling, agitation, and headspace, leading to defined media oxygenation. Now this company of engineers, it turned out, also made these things. These things are called workstations, and they provide a continuous gas-controlled environment. So now we can grow our cells continuously put our hands in the front here, handle our cells, move our cells within this gas-controlled workstation. So we can have this at our physiologically normoxic setting. We can control our media. So now we've got a holistic approach. We're covering the media, we're covering the headspace, we're covering the handling. Okay, so let's switch gears back to these images that we showed you right at the very beginning. Here's our mesenchymal stem cells. Okay, mesenchymal stem cells are sourced from bone marrow. Bone marrow is kind of jam in its constituency. We seed it out into flasks. We recover these little splodges of cells, and we can zoom in on these little splodges further and further and further, and we see that they're a relatively homogeneous cell population. They look quite beautiful, actually. Now these cells are characterized by their ability to form cell types that are representative of bone, fat, and cartilage. Okay? Canonically, that's their profile. They're also in current cell therapy application globally for over 450 active recruiting clinical trials. They're part of that $30 billion industry that we heard about earlier. Okay? Some are in clinical trials, some have made it into marketplace. Okay? That are sold, that are on prescription, essentially. They were invented by this guy back in 1976, and this guy was Alexander Friedenstein, and Alexander Friedenstein was at the USSR Medical Research Institute. And what he did was he took these mesenchymal stem cells, he isolated them, and he popped them into little cassettes, which he showed were then able to form bone and cartilage. So these are a bone marrow cell which can give rise to musculoskeletal tissues. Okay? And these are the cells that we're going to work with and describe over the remainder of the presentation. The mesenchymal stem cell niche, it might not surprise you to learn at this point, also has a low physiologically normoxic value. Now the mesenchymal stem cell niche doubles as the hematopoietic stem cell niche, and hematopoietic stem cells are the cells that give you all of the cells that you find in your blood. Okay? All of your blood cells come from hematopoietic stem cells. And this niche has a low PO2, a low physiologically normoxic value. So what we sought to do was now we've got the conditions, now we can control everything that we want to. So now we can control what's going in our media. We can control our handling, so the cells are never exposed to air oxygen whatsoever. And we can compare it to one of our incubators. This is an incubator that has nitrogen introduced to make the oxygen physiologically relevant. And we can now do simple experiments and count the number of cells that we isolate from bone marrow. And what this shows us, this is our workstation. This is our all-in approach, our holistic approach. What this shows us is that actually when we use our workstation now, our continuously contained system, we get substantially improved cell numbers over our next best available technology. More cells equals better ability to treat patients, okay, in its simplest essence. Now, we're just getting going with this, okay? The wheels are just starting to gain some traction. Of those 450 clinical trials that we told you are underway with these cells, none of those cells use physiological normoxia in any extent, shape, size, or purpose, not one, because the technology is only just getting up to speed, because we're at the coal face here. We're at the coal face. Okay, so physiological normoxia, that's the concept we've tried to bring. From the bottom to the top of stem cell biology. So how much does this matter? How far does this go into the way these cells click? And I'm going to give you some very up-to-date data provided by a very bright student, Rakad, who should be hopefully somewhere in the audience. And what we want to know is whether physiological normoxia has a role on DNA, on RNA, and protein. DNA is the stuff that gives rise to what we are. All of our cells have a DNA nucleus. Our DNA nucleus gives rise to RNA, and our RNA is then turned into protein, and protein is what we're all made of. Okay? Every bit of us is protein in essence. So what Rakad's been able to show was that actually, so what he's measuring here, he's measuring a mark that happens on DNA. And what he's showing here, and this is our air cultured cells, and this is our workstation cultured cell in the red bar, and this is a midpoint in the black bar. What he's showing a reduction in DNA marks when cells are cultured in that 2% controlled condition. And the less marks means that you get better control of your RNA. Okay, your RNA is now more accurately being turned in, your DNA is now being more accurately turned into RNA. He's also been able to show that the genes which control these marks are also switched off. Okay, they're down-regulated. That's these guys here and these guys here. So the genes that control these levels of DNA marks are being switched off. And he's also been able to show that the protein that controls whether or not these DNA marks happen or not are also switched off. So by just taking these cells, these bone marrow mesenchymal stem cells, and by reducing the oxygen, culturing them in a physiologically normoxic condition, he's imposing changes on the DNA, he's imposing changes on the RNA, and he's imposing changes on the protein. So physiological normoxia, from the bottom to the top of stem cell biology, does it impact on DNA? The answer is yes. Does it impact on RNA? The answer is yes. Does it impact on protein? The answer is yes. This is imposing and impacting on all facets of cell biology that we are able to look at. Now, the thing speaks for itself. I gave a keynote at King's last week or 2 weeks ago, and I kind of cheekily finished on this slide and then invited questions, but I'm not going to do that today. But in terms of a summary, what we would say is that physiological normoxia is an underexplored and yet incredibly important aspect of the cell culture milieu. Historical lessons define future directions. Continual improvements in knowledge access will drive forward 21st century science. The stories that lie in the past, and this is the back to the future bit, inform the directions we go in the future, and we ignore them at our peril. Simple work has been performed. Simple work that wasn't accessible to us even a decade ago, we can now access as digitization proceeds and everything gets up to speed. And what that will lead to, these improved in vitro models where we can model physiology, will drive forward fidelity of our in vivo understanding. Okay, so at that point, that's the science bit finished. I was just going to quickly move on to the bio bit, but we heard about that already, so I'm going to skip over that. Thank you, Mark. We've heard about this. This is the teaching stuff that I do, so we'll skip over that. The citizen bit, I established the Human Tissue Authority approved regulatory framework for Keele shortly after moving here. I serve as the biological safety advisor. I'm proud to sit on Senate and Council and continue to act in those roles. I thought it might be nice just to as I wrote this, I wrote a career in numbers, and then I had to do a little bit of a reality check to myself. Well, I thought, now, whilst I may be 45 in September, which puts me 5 years away from qualifying for a SAGA holiday, I thought a career in numbers might be a bit premature. So a mid-career, okay? Publications around 80, citations around 1,500, patents 3, PhD students graduated 9, 5 current. One has graduated since I gave Mark his numbers, so that number has changed. Research grant income, healthy. Guest possessions in China and Vienna, and editorial boards, which is serving the greater scientific community. Acknowledgements. We're on our third last slide. It's important to acknowledge current and previous members of my research team. I'm very guilty of always not communicating the whole picture. The whole picture lies somewhere in this grey matter, and people learn to have faith in me quite quickly. It does all hang together, honestly. Funders, science doesn't happen without funding support, and so we'd always encourage everybody to be nice to funders and give to charity. Professor El Haj, Professor Monica Spiteri, Professor Bill Farrell, who've all provided support, insight, comradeship, and all those key things, and mentorship over the duration. I would never have acknowledged having a mentor until recently, but I now accept that I quite possibly did. They were just cleverer than me, and they didn't call themselves my mentor. And Professor Jim Elder, and Jim Elder's here essentially because of him introducing me into the North Staffs Medical Institute, basically on the basis that I had a Scottish accent. to all extents and purposes. But I hope I've served him well on that point. He recognised a good work ethic in the mother tongue, as he described it. Former and current colleagues at the Beatson, the UTSW Medical Centre, Roslyn and Keele. Key mentors along the way, Ken Parkinson, who taught me how to work, Jerry, who taught me to look for the angles, Woody, who taught me how to think, and Jim, I think, who nurtured independence, actually. Long-term industrial collaborators at Baker Ruskin. We've been working with them for 8 years. They've licensed patents, and we've got a great relationship. And of course, family, mum, dad, granny, who's approaching her century, who can't make it for that reason. Friends and Anne. And actually, I found this quote in my PhD thesis, which I think is as relevant then as it is now, and that was in 1999. I went for the adventures we've had and are yet to have, and I think we continue to have adventures. We continue to move forward in research and in life, and without the support that's provided, these things wouldn't always happen. Now, so second last slide. So the question then is, how do we get these little flower children here from this early 1980s picture to become this? And then how do we get these bright young academics? This is my current research group here. How do we get these bright young new academics to ask the right questions and to keep driving forward and to interrogate science and to move forward in that manner? And it wouldn't be right to have a Scotsman give a talk without a picture of a kilt. So here's the kilt. And where do these guys go? Where do they end up? And I think the future is bright. I think as information technology really takes hold, and as we move forward in accessibility and the ability to get whatever information we want, I mean, these guys here had no idea there would be a World Wide Web. They had no idea they would have smartphones. These guys here take that in their stride. So where these guys will be in 40 years, I think, will be frightening, and we would not recognise it if it was set in front of us. And I do think the future is incredibly bright. So with the theme that I've kind of tried to introduce, this Back to the Future theme, we'll finish with this quotation, and thank you for your attention. Thank you.